Hey y'all, we're Group 13 and we did our video over elephant population genetics and conservation, so let's go.
Cool. So the Skittles represent the African elephants and the M&Ms represent the forest elephants. And now we're going to scatter them. Scattered like this, they kind of look like one species, but conservation-wise, that's actually not very good, because if I come in and represent some natural disaster and just take out this bunch, and then we look at them, if you lump them together like that and say they're one, then that undermines how vulnerable the one species actually is.
This is why genetics is so important to tracking animals. Instead of just tracking the entire group and maybe leaving out some unprotected individuals, you can be very specific about what you're looking for.
mtDNA or mitochondrial DNA convert chemical energy from food to ATP. mtDNA is passed only from mothers to their offspring with few mutations, and their mtDNA is not usually dispersed geographically. In contrast, male elephants disperse from their natal herds when they reach maturity and wander in search of females. Female elephants come into estrus for only a few days every few years, so roving males use their keen hearing and smell to find them. Because both males and females contribute to DNA from each cell's nucleus, males that breed with females far from their kin promote gene flow.
So since male elephants directly affect gene flow, we can track this by using genetic markers such as microsatellites.
The scientists used genetic markers called microsatellites to measure genetic variation in specific segments of nuclear DNA. They examined the markers to determine how genetically distinct the elephants from various sample locations were from each other. These are the normal sequences, and then this is our tandem sequence.
So we're using these colored beads to represent our cytosine, guanine, thymine, and adenine, respectively. You can actually pick them out, and they're like patterns and markers in the genomes. So the one for the elephants that they found is ATAG. I'm building the ATAG microsatellite marking sequence. So that repeated with tandem, this is what that would look like, and compare that to a randomly made other sequence.
So when researchers looked at all of this information, they were figuring out that forest elephants were actually becoming more endangered than other species of elephants because they were getting lumped in with all of the other species of elephants. And actually, for conservation purposes, that just doesn't work. So what they needed to do was actually be more specific and understand that different species have different needs. So, by using population genetics and tracking migration patterns, researchers were able to use those to their benefit and say that the best way to conserve elephants is to actually split them up by species and by regions, because that'll give them the amount of protection that they actually need.